We will now invite Dr. Kiran Thapaliya, who is a research fellow at the National Centre for Neuroimmunology and Emerging Diseases at Griffith University, Gold Coast. Kiran and I have worked together in a previous life. A lot of Kiran's work focuses on the use of advanced multimodal MRI techniques to develop novel neuroimaging methods. His particular aims for ME and long COVID are to help enhance diagnostic precision and our understanding of the neuroimaging and neurological changes in this condition. So please welcome Kiran.
Okay, thank you, Stanley, for the introduction. Yes, we are missing you here. Yes, I'm Kiran, working as a research fellow at the National Centre for Neuroimmunology and Emerging Diseases, NCNED, Griffith University at Gold Coast campus. Today, I'll be presenting most of our research update on ME and long COVID. In today's presentation, I'm going to share different research projects findings, including quality of life in ME and long COVID, TRP channels dysfunction in ME and long COVID, immune exhaustion in ME and long COVID, and definitely MRI findings in ME and long COVID. So I would like to thank our funding buddies, including NHMRC, Stafford Fox Medical Research Foundation, ME Research UK, and other funding bodies here. This wouldn't be possible without the funding from these agencies or funding bodies. And I would like to thank every participants who have been directly or indirectly involved in our research project. And this wouldn't be possible with their participation and support for this research. I'm pretty sure now most of the presenters over here are well known about the ME. But if someone wants to watch the video recorded later on, I would like to give a brief introduction about the ME. ME is a complex illness that affects multiple body systems. If we look at the figure here on the right-hand side, we can see that ME affects the multiple parts of the body, including the brain, respiratory systems, and it has the irritable bowel disease-like syndrome, and it has the different joint pain and the muscle pain. In 1980s, World Health Organization defined ME as a neurological disorder, and people suffering from ME experience range of symptoms, including post-exertional malaise, cognitive dysfunctions, sleep disturbance, unrefreshing sleep, pain, and flu-like symptoms. They also have the cardiovascular problem as well as gastrointestinal upset. More than 24 million people suffer from ME, but unfortunately, we don't have any diagnostic test. And diagnostic is mainly based on the clinical case definition, including Canadian consensus criteria, known as CCC, and international consensus criteria, ICC. And even we don't have the evidence-based treatment for ME. As we know, in 2020 to date, more than 775 million people have suffered from COVID-19, and it has been reported that at least 10% of COVID-19 infected people develop themselves into long COVID. And according to the WHO definition, long COVID is defined as the development of new symptoms 3 months after the initial infection, which persists for at least 2 months with no other explanation. And as we know that research studies have shown that symptoms between the ME and long COVID are very much similar. And if we look at this table here, we can see that out of the 25 symptoms listed on this table, at least 20 symptoms overlap between the ME and long COVID. And it has also been shown that 13% of long COVID patients meet ME criteria. And as shown earlier, they exhibit the similar symptoms, and 70% of ME patients report post-infectious onset. So if that's the case, what happens to the quality of life of health-related outcomes in ME and long COVID. The study led by Miss Brenna Weigel, she studied the quality of life in ME and long COVID. From 60, she recruited 60 ME people and 31 post-COVID condition, which is also known as long COVID, and 54 healthy control. And she used the 36 item short form health survey, which is known as SF36, and Old Health Organization Disability Assessment Schedule version, which is known as OHDAS, to see the health-related outcomes in ME and long COVID. And we didn't find any significant difference in ME and PCC. That means the SF36 and OHDAS score were significantly reduced in ME and PCC when we compare to the healthy controls, but they were similar in ME and PCC in the overall long COVID. And we also extracted the symptom and severity scores derived from the Canadian Consensus Criteria CCC and ICC. And out of those other symptoms, we found that lightheadedness and unrefreshing sleep were only significantly different between people with ME and people with post-COVID condition, which is again like long COVID. So this shows that the quality of life is significantly impaired in ME and long COVID when we compare to the healthy controls, but they were similar in ME and PCC. And another study led by one of the research fellows here, Dr. Natalie Eaton-Fitz, she looked at the immune exhaustion in ME, long COVID, and healthy control in 14 ME, 15 long COVID, and 18 healthy control. And she looked at the different genes here, and it's been reported that 5 genes were significantly upregulated and 9 genes were significantly downregulated in ME. When we compare to the healthy control, the graph here in the right-hand side shows the downregulated genes and the upregulated genes in ME when compared to the healthy controls. Similarly, when we compare the different genes in long COVID and healthy control, again, 15 genes were significantly upregulated and 14 genes were significantly downregulated in long COVID when compared with the healthy controls. And out of different genes, 7 genes were overlapped between ME/CFS and long COVID, suggesting the involvement of similar molecular pathway involved in the immune dysfunction in ME/CFS and long COVID. Then we look at the TRPM3 dysfunction. That what is TRPM3 dysfunction? I'm going to say what this TRPM3 is. So TRPM3 dysfunction, TRPM3 also known as transition receptor potential melastrin free. It belongs to the family of TRP channels. Mostly it is involved in the passing of the calcium into the cell. That means if TRPM3 is dysfunction, the calcium cannot pass into the cell and there might be the cellular impairment and the cell might not function properly. So to test this TRPM3 function in ME/CFS, we use the whole cell patch clamp technique, and this is the gold standard technique for determining the receptor function. And here we recruited the 6 ME and 6 healthy controls here, and we measure the TRPM3 function in healthy control and ME, and we found that TRPM3 function in ME is significantly reduced when we compare to the healthy controls. So now, if TRPM3 is dysfunction in ME, can we restore the function of TRPM3 in ME or not? So in vitro experiment was conducted at ME and at healthy control, and when the naltrexone was treated in ME cell, we found that the naltrexone restored the function of TRPM3, and there is no significant difference in TRPM3 function between ME and healthy controls. If you look at in the bar graph here, there is no significant difference. Then we look at a treatment target for ME, because in vitro experiment, we have we showed that natroxone restored the function of TRPM3 channel. Then we tested on the 9ME taking LDN and 9 healthy control, and we found that TRPM3 function was similar to the healthy control here. If you look at this bar graph here, the white one is the healthy control and the black bar is ME, and we found that after people taking LDN, their TRPM3 function is restored. That means low dose naproxen helped to restore the function of TRPM3 in ME patients, which could be because after taking the LDN, the more calcium could pass into the cell, and the cell has started functioning smoothly. So now we have shown the TRPM3 channel dysfunction in ME, and low dose naproxen has restored the TRPM3 function in ME. So what about in the long COVID now? So here we perform this experiment in 5 healthy control. This study was led by another research fellow here at NCNED, Dr. Etienne Martinez-Sasso. So we recruited 5 healthy control, 5 ME and 5 long COVID. And if we look at the graph here, again, in x-axis the healthy control, long COVID and ME, we can see that in healthy control and the long COVID, TRPM3 function is reduced, whereas when we compare the LC and ME, they are very much similar. So that means that it shows that the TRPM3 function is significantly reduced in healthy control significantly reduced in long COVID when we compare to the healthy control, but this is also similar in long COVID and ME. So if that's the case, can we restore the TRPM3 function in long COVID? So we here, again, the study led by Sasso, we recruited 9 healthy control, 9 long COVID without taking LDN, and 9 long COVID who are on LDN. And when we compare the TRPM3 current amplitude here, we found that between the healthy control and long COVID taking LDN, the TRPM3 function is similar. That means there is no significant difference in TRPM3 function. That means LDN has restored the TRPM3 function in long COVID people who was taking LDN, but there are significantly again difference in long COVID who was not taking LDN. So far, what I have shown here, what NCNED have found is that the quality of life is impaired, immune exhaustion in ME and long COVID, and TRPM3 dysfunction in both ME and long COVID, and LDN has restored the TRPM3 function in both ME and long COVID conditions. Now, I would like to show some of the NCNED findings, MRI findings here. So we use the 7 Tesla MRI. So most of our MRI findings here are from 7 Tesla MRI. So why we choose 7 Tesla MRI is because it provides better quality images. It improves the visibility of pathological alteration that might not be detected in 3 Tesla MRI or 1.5 Tesla MRI. And it provides a clear depiction of anatomical details. If you look at the figure here, the left-hand side is 3 Tesla MRI, structural MRI. Here it is in right-hand side is a structural MRI acquired from the 7 Tesla MRI. And you can see the most of the white matter regions are very much clear in 7 Tesla MRI when we compare to the 3 Tesla MRI. So the first study that we conducted with 7 Tesla MRI is to see the functional connectivity. Now, I'll try to explain and try to be very slow to make sure that what does this mean. So here to measure the connectivity between the 2 regions, that means we have, for example, let's say we want to see the connectivity between the 2 brain regions. What does the connectivity means? It measures the similarity between brain signals arising from the 2 regions. And we use the functional MRI here. The functional MRI, it tracks the blood flow in brain to see which areas are most active and compare it to what we are doing at that time. For example, Let's say you are tapping the finger on the disk, right? And when you are tapping the finger on the disk, we want to identify when you are tapping which brain regions are activated, or which regions are connected or sending the signal to each other region. So we identify whether those connections between two regions are weaker or stronger in MECFS patient. So here we use the Stroop color task, which is a cognitive task. So here the take-home message from here is like we want to see the brain activations regions when you are performing some kind of task. So in this experiment, we acquire the data from 31 ME and 15 healthy controls. And we use the Stroop task here. So actually what we do here is we put the participants inside the 7 Tesla MRI scanner. We give them button to press. One button is for yes if answer is right, and another button is for no when the answer is wrong. So we use the cognitive task known as the Stroop task here. So when the participant is inside the scanner, this message pops out. It says that the color of the upper word corresponds with the meaning of the lower word, and if it matches, participant click the button for yes, if not, another button for no. This is an example of the task here. So this is it says the upper word is blue in color, and it says the blue, the participant clicks the S button. But if we look at the middle here, it reads blue, upper or reads blue, and it's in green in color, and it's in green. So participants get confused here because they read only blue and they try to click the no button here. But this is exactly, it is the S button, it's called incongruent stimuli, and they get confused over here. And this is where we want to measure to see which brain regions are activated when they are performing these tasks. That's why we use the functional MRI to see the brain activations when they are performing this task. So when the people are performing this task, and we found that the functional connectivity was significantly reduced in ME when we compare to the healthy controls. And these two regions, one is the brainstem region and then is the cerebellum region. The connection between the cerebellum and the brainstem was significantly reduced in ME when we compare to the healthy controls. So now, as mentioned earlier, ME and long COVID, their symptoms are pretty much overlapped. So if the symptoms are overlapped, what about the brain alterations? Do they have the similar brain alteration or not? So for this one, we use the structural images from the 7 Tesla MRI. We recruited the 30 ME, 15 long COVID, and 15 healthy control. And in this case, we use the hippocampus as our test region because hippocampus is involved in spatial learning, working memory, and recognition memory. And as we all know that ME and long COVID has suffered from the cognitive impairment, we want to see is there any alterations in the hippocampus volume or not. Let's look at the findings here. We found that if we look at this figure in the left-hand side, the y-axis is volume of the regions given in millimeter cubed, and x-axis is the different cohorts. That's the ME, long COVID, and healthy control. So the red one is ME, green one is healthy control, and blue one is, sorry, red one is ME, green one is long COVID, and the blue one is healthy control. And we found that left subiculum head was significantly larger in ME and long COVID when we compare to the healthy controls. But when we compare in ME and long COVID, they were similar. Similarly, if we look at another region called left pre-subiculum head, they were significantly larger in ME and long COVID when we compare to the healthy controls. Now the question is, what could be the reason that why we are seeing the larger hippocampus volume in ME and long COVID? this could be because there might be some neurogenesis happening in the hippocampus. So what happened is that this neurogenesis could also be due to the environmental factors and stress that trigger the ME/CFS symptoms. And we did not find any significant differences in the hippocampus soft fluid volumes between ME and long COVID. And this is very much, this also indicates that there is some kind of overlapping between these two conditions. Then we correlated the hippocampus volume with a pain score. Here again, if you look at in the y-axis, this is the pain volume. Volume of hippocampus soft field volume given in millimeter cube, and this is the pain score severity. Here it means like if you have the low pain score, this is the higher severity, and if the higher pain score means there is no pain. Okay, so the higher this, if it's closer to 100, no pain. If it's closer to 0, it's higher pain. So if we look at here in ME, we found that as your pain level is higher, you have got the lower volume, right? So this was reported in left presubiculum head volume, and same with the left femoria. If we see that, if you have the higher pain intensity, you have got the lower volume. And let's look at in the long COVID. Here we can see if you have the higher pain level, you have got the higher volume. Similarly, in the right femoria, we've also found that we have a higher pain, higher volume. So this is pretty much interesting. So you are saying, so hippocampus, because why the hippocampus soft field volume or hippocampus is related to the pain is that in hippocampus receives the pain from the different connectivity. For example, corticolimbic connectivity is there, which integrates the pain into the hippocampus. But there is a contrasting relationship between the pain in long COVID and ME. This contrasting relationship between hippocampus soft field volumes and with the pain could be attributed to more severe symptoms of ME/CFS, because the average symptom duration of illness in ME is almost like 13 years here, but whereas in the long COVID, it's just like 2 years. So maybe we need to identify whether they might be similar in the future or not. We need to perform the longitudinal study. But what another study has shown is that people with PCS, that is a post-COVID condition, with ME/CFS, their pain intensity was very higher when we compare with the people only with the long COVID or the PCC condition. So, but as we said, like we need to do the longitudinal study to see whether we'll get the similar correlation in the hippocampus volume and the pain in the future between the ME and long COVID. So after the structural one, we want to see what happens to the brain neurochemicals in ME/CFS, in ME and long COVID. So here we choose the posterior cingulate cortex as a test region. Why we choose this region is that this region has 40% higher metabolic rate than the other brain region. That's why we say, okay, if that's the case, what's happening with the brain neurochemicals here in this region in ME and long COVID? So for this experiment, we recruited 17 ME, 17 long COVID, and 10 healthy controls, and try to estimate the different brain neurochemicals like glutamate, glutamine, and acetyl aspartate, because these neurochemicals are heavily expressed in the brain regions. And we're gonna see, okay, what's happening with these neurochemicals in this region. And here what we found was that glutamate was significantly higher in ME and long COVID when we compare to the healthy controls. If you look at this figure here, again, the y-axis is the glutamate level, and this is the different cohorts. Again, the red is in ME, green is long COVID, and the blue is healthy controls. And we found that glutamate level was significantly higher in ME and long COVID, again, compared to the healthy controls. But this brain glutamate was very much similar, again, in ME and long COVID. There was no significant difference. So if we looked at the N-acetyl-aspartate, we found only significant differences in long COVID, which was very much higher. Na level was very much significantly higher in long COVID, again, when we compare to the healthy controls. Now, what could be the reasons why we are seeing higher amount of glutamate in ME and long COVID? One of the potential reasons is that there is some kind of inflammatory cytokines happening in the ME, which have been shown by the different studies. And these inflammatory cytokines affect the glutamate release, transmission, and metabolism, leading to higher concentration of glutamate in the brain. So that could be the reasons behind that why we are seeing the higher concentration or higher level of glutamate in ME and long COVID. So in conclusion, what we have found so far is that quality of life is impaired. There is the immune exhaustion and dysfunction of TRPM3 and brain dysfunction of brain as well as TRPM3 in ME and long COVID. TRPM3 and brain dysfunction were similar in ME and long COVID conditions, providing further evidence for a significant link between the two conditions. LDN restored the TRPM3 function in both ME and long COVID, indicating potential future therapeutic treatment for both conditions. These findings may help to better understand the pathomechanisms of ME and long COVID conditions. And this is NCNED team led by Professor Sonia Marcel-Gradisnik. And as Daniel mentioned, we are also looking for the participants, long COVID and ME for different MRI studies and the blood donations. If you are interested in taking part, please contact at ncned@griffith.edu.au. And we are also organizing a conference for ME and long COVID this November. And if any one of you is interested in taking part or submitting an abstract, please feel free to contact the below email. And we are also running the clinical trial for long COVID using LDN, and the clinical trial for ME will come in soon. If you are again interested, please email at ncned@griffith.edu.au. Thank you so much again. I'm happy to take questions now.
All right. Thank you so much, Kiran, for sharing an overview of NCNED's work with a particular focus on the neuroimaging that you've been working on as well. Same as before, does everyone or anyone have a question for Kiran? Otherwise, I've accidentally sent to the group chat that I can read out in the meantime. My first question, Kiran, was with the changes reported in the gene expression profiles and TRPM3 dysfunction, I guess what Daniel suggested in his first or in his presentation was the strength of their research as they are trying to narrow down the signature that's relevant to ME and what may be overlapping with other conditions. So is there any data to show that these changes are highly specific to ME and long COVID? How do they compare to other inflammatory conditions, or is it just reflecting inflammation?
Yeah, this is a really good question, Stanley. We have done this gene and TRPM3 dysfunction in, I think we did publish the TRPM3 dysfunction in Gulf War illness and GWI condition. I'm not sure about the gene expression, but yes, I think potentially we'll be looking to other condition and see how it differs with ME/CFS and with ME and long COVID and other condition. But yes, we are working with the GWI conditions too, which we have already published in, I think, this year. So yeah, hopefully we'll do in the other condition too.
Yeah, good, thank you. Could I see clinically things like LDN being used for a few rheumatological conditions and helping dampen inflammation as well? So whether that's what we're seeing with some of these study findings. Daniel has a question.
Hey, Kiran, nice talk. So with the TRPM3 stuff, was that
Mhm.
all in NK cells?
Yes.
Is there any plan to look at um, any other aspects of calcium homeostasis in NK cells and accompany that with, say, cytotoxicity assays or other things that you guys might think might be going wrong and see what the relationship there might be in terms of um, altered calcium homeostasis and altered effector function by the cells potentially.
Yes. Daniel, I think I mostly work with the MRI with the
I know
neuroimaging
you're the
part. So
MRI guy,
yeah, so
yeah.
uh to be honest, I don't know. But I think if you have any questions, I think I'm pretty sure I think the cytotoxicity has been shown in ME, but in terms of TPM3, other looking at the calcium homeostasis, I think the best person to answer this question is Professor Sonia Masakarisnik and other projects happening with the calcium thing.
Yeah.
Happy to
All
forward
right.
your thing to her if you
Yeah.
want.
No, just that our lab does a lot of calcium homeostasis work, so I always find it interesting. So
Mm, thank you.
there's plenty you can dig into there.
Yes, yes.
Thank you. Any other questions for Kiran? I've got one specific to MRI and neuroimaging.
Perfect. That's wonderful.
um, so the reason for this question is we know the central nervous system includes the brain and the spinal cord. So
Mm,
one of my questions was, again, in a lot of chronic conditions, there's things like peripheral and central sensitization to things like pain or itch.
mm.
That causes a lot of symptom burden for patients. Has there been any studies that you're aware of in people with ME or long COVID looking specifically at the spinal column? The reason for this question is there any features of dysfunction in the afferent or efferent signaling in the spinal cord, or is it mainly just integration centrally in the brain that's being disrupted?
Yeah, I think this is really good question you raised, Stanley. But what we have seen in ME as well as in long COVID is there are different tracks in the brainstem which connects central nervous system with the cortical regions and pass that signals to the spinal cord. And we have shown those tracks are impaired in ME and long COVID. And these tracks are heavily involved in the pain also and in the cardiovascular or respiratory system also. So I believe that if we look at those tracks that connects between the brain and the spinal cord, which we have shown actually in ME, but we haven't done specific to spinal cord. However, there are some white matter alteration in spinal cord too, which is pretty much what we have found in the brain. So there is the potential reasons to connect those things, but we haven't got any data of spinal cord to play in ME so far. But hopefully in the future, we'll do it. And it's very tricky also. It's not straightforward because then you have to restrict so many things during a scanning session. And yes, but definitely, I think there is a huge potential to look signals passing from the brain to the spinal cord.
Yeah, feasibility is a big consideration in any research. And I guess to summarize what you've said, there's changes both in the tracks and the central processing
Mm.
areas in ME and long COVID. Okay. Any other questions? Otherwise, I've got another one.
Yeah.
Are there are there any particular signatures you've identified for ME and long COVID that are quite stable despite clinical variability? Again, I guess going back to a similar question I asked Daniel, what's a to date relatively consistent feature we see across different clinical manifestations of ME?
Yes, I think what we have shown here is continuously like we since 2016, Stanley, MRI data that we, and the 2021 data from the new projects with the different ME cohort, what we have found is that one is the brainstem connectivity. There is something happening with the brainstem. Definitely we can say that, there's something in the brainstem in ME that we have seen continuously impaired. But again, as I said, there are different nuclei, different tracks. And it's very hard to pinpoint like which tracks and which nuclei are dysfunction. But definitely we are seeing that in the brainstem. And another one is the hippocampus. In data from a different cohort in 2016, we saw larger left subiculum head volume, that is the hippocampus volume, was significantly increased in ME. I think we acquired that from the 45 ME CFS and 27 healthy controls. And we saw that it was significantly increased, right? Then that was from the 3T MRI. Then again, in 2021 study, from the 30 ME CFS and 15 healthy control, we found the same regions or increased in ME and long COVID. So it's that regions is popping out every time. And this is unique to other condition because hippocampus volume, if you look at the Alzheimer's and other diseases and other condition, almost like most of the paper, they have reported decrease in the hippocampus volume. But here in ME, we are seeing the increase. It's very hard to say why, what is the clinical manifestation or what's the clinical reason behind it, but we are seeing the increase in different cohorts or using the different MRI techniques. So we believe that this could be one of the key reason in ME. That's why the people are suffering from cognitive impairment. But still, we need to track those, longitudinal study and see whether that regions is impaired at different time points. But yes, and in addition to that, we have also shown the TRPM3 channel dysfunction continuously in ME, and those TRP channels are also located in hippocampus and brainstem regions. So this is very good, I would say, the complementary findings for each other because TRP channels are dysfunction and they are located in different brain regions. So that could be like, maybe the TRP channels are dysfunction. That's why we are seeing changes in the hippocampus or the other brain regions. Or, it's very hard to say, but, could be one factors could be the TRP channels. But as to summarize in one point, yes, we are seeing some unique signatures in ME, especially in the hippocampus and brainstem using the MRI.
Thank you.